Okay, today we'll start hepatobiliary system. This is in anatomy. Have you read about the hepatobiliary system?
No, sir.
No. Okay. In physiology, we'll study something about anatomical physiology. Anatomical physiology of hepatobiliary system. In this topic, though this is a small topic, one or two question is always asked. So it is easy to prepare it, and uh, most of the easy questions will be asked from this hepatobiliary system. So you need not to worry about it. Every topic will be covered. Okay, let's start about hepatobiliary system. What do you mean by hepatobiliary? Hepatobiliary means, hepato means liver, biliary means bile. So main function of liver is secretion of bile. And do you know the function of bile?
Emulsification of fat.
One, yes, it is the emulsification of fat. Emulsification simply means breakdown of fat. Breakdown of fat into fatty acid and glycerol. One of the important question that is always asked in your exam is the function of liver. One of the important question that will be asked in the examination is the function of liver. So simply by understanding, you can jot down all the important function of the liver. Before we study the function of liver, let us study about the functional anatomy of liver. You can see that this is the cross section. This is the cut section of the liver. This is the cut section of the liver. To understand the function of liver, to understand the function of liver, you should first know the functional anatomy of liver. If you know the functional anatomy of liver, then you can easily describe, or you can easily write down the function of liver. So let us see in this diagram that you can see that. Can we have basic idea about liver? Liver is the largest organ. Liver is the largest organ of the body, and its weight is 1.5 kilogram in human. It is the largest organ of the body, and its weight is 1.5 kg in human. And do you know the functional unit of liver? What is the functional unit of liver? It is the liver lobule. Functional unit of liver is liver lobule. Functional unit of liver is liver lobule, and you can see that this is a liver lobule. Liver lobule is a cylindrical in shape. Liver lobule is a cylindrical in shape, and it is arranged around the central vein. Functional unit of liver is the liver lobule, and it is arranged around the central, and this central vein, it opens into the hepatic vein, and liver lobule is a cylindrical in shape, and again this liver lobule it is made up, and you can see liver cell plate is bile canaliculi. Between the liver cell plate is a bile canaliculi. In this bile canaliculi, hepatocytes or liver cell plate, it will produce bile, and it is secreted into this bile canaliculi. And from this bile canaliculi, it opens into the bile duct in the bile. It is open into the bile duct. Again, I am repeating it. Before studying the function of liver, let us study about the functional anatomy of liver. I have already said that functional unit of liver is liver lobule. Functional unit of liver is liver lobule, and it is a cylindrical in structure. It is a cylindrical in structure, and it is arranged around the central vein. This central vein, it opens into the hepatic vein, and hepatic vein, it again drain into the inferior vena cava. And let's see about the functional unit of liver is liver lobule. Liver lobule is again made up of liver cell plates. Liver lobule is made up of liver cell plates or hepatocytes. And between these two liver cell plate. Between these two liver cell plate is a bile canaliculi. In this bile canaliculi, bile is secreted from these hepatocytes. Bile is secreted from these hepatocytes and it is poured into this bile canaliculi. From the bile canaliculi, it opens into the bile duct in the fibrous septa of liver. Okay. Now again, other structure is this is the portal vein. This is the portal vein. Portal vein. It will drain or it will collect blood. It will collect blood from the GIT. It will collect blood from the GIT venous blood. GIT venous blood is collected by the portal vein. GIT GIT venous blood. GIT venous blood is collected by the portal vein, and from this portal vein, portal venules, it opens into the liver sinusoids. Again, I'm telling it. Venous blood is collected. Venous blood from GIT is collected by the portal vein, and it opens into the liver sinusoids, So in this liver sinusoid, it is mainly the venous blood from the GIT. And in this liver sinusoids, it is lined by special type of cells. That is endothelial cells, typical endothelial cells. It is lined by the typical endothelial cell. And it is also lined by the macrophage. That is Kupffer cell. It lined the liver sinusoids. Again, just let's see it. Blood collected from the venous blood collected from the GIT by the portal vein, and it opens into the liver sinusoids. And this liver sinusoids is lined by typical endothelial cells and Kupffer cell. Kupffer cell is a resident macrophage. In the liver sinusoids. And function of Kupffer cell is to engulf bacteria. And foreign matter. And from this liver sinusoids, it drain into the central vein. From central vein, it drain into the hepatic vein. From hepatic vein to the inferior vena cava. And you can see that hepatic artery, it also supply the fibrous septa. And liver sinusoids. This hepatic artery, it supplies the liver septa and liver sinusoids. So in this liver sinusoids, mainly the impure blood, it is drained into these hepatic sinusoids. And from hepatic sinusoids, it ultimately drain into the central vein. From central vein to the hepatic vein, and from hepatic vein to the inferior vena cava. Now, you can see that from this diagram, you can see that between the endothelial cells or hepatic sinusoids and the hepatic cells, there is a space. Called space of Deschi. A space of Deschi is a space between liver sinusoids and the hepatocyte. A space is called a space of Deschi. In this space of Deschi, extra amount of fluid is collected and it is drained into this lymphatic duct. So in the liver, when there is extra amount of fluid is collected, then from this duct, it is opened into the lymphatic duct, and through this lymphatic duct, it is drained through the thoracic duct. So do you understand the anatomical function of liver? I am just again repeating it. Functional unit of liver is called liver lobule. This liver lobule is a cylindrical in structure and it is arranged around the central vein. And you can see that this liver lobule is made up of liver cell plates, and between the two cell plate is a bile canaliculi. In this bile canaliculi, these hepatocytes, it will produce bile and it is poured into this bile canaliculi. And from this bile canaliculi, it is opened into the bile duct And the venous blood venous blood that is collected, venous blood from the GIT, venous blood from the GIT, it is collected by the portal vein, and it also opens into this liver sinusoids. And this sinusoids is lined by this liver sinusoids is lined by the special type of cells, that is typical endothelial cells, and it is also lined by the Kupffer cell. And function of Kupffer cell is engulfment of bacteria and foreign matter. These endothelial cells have large pores, so the blood in this venous blood, it can easily filter into this space of DC. Space of DC is the space between these sinusoids and the hepatic cell, and it ultimately opens into this lymphatic duct. So this is the functional anatomy of liver. Now let us study about the function of liver, because in your exam it will be asked about write down the function of liver. So this is the structure of liver. In this diagram you can see that blood flowing in one direction and bile is flowing in other direction. This is counter current. So if you are asked in your exam for 1 marks, write down the function of liver, just enumerate the function of liver. No need to explain the function of liver. So one of the primary function of liver is the production of bile. Another function of liver is the liver function as a reservoir of blood. Because large quantity of blood can be stored in its blood vessel, that is liver sinusoids and the hepatic vein. About 0.5 to 1 liter of blood can be stored in liver sinusoids and hepatic vein, especially when right atrium, especially when there is high blood pressure in the right atrium, then there will be backflow or back pressure of blood, and there will be accumulation of blood in the liver, and liver can store up to 0.5 to 1 liter of blood is stored in the hepatic sinusoids and the hepatic veins. So first function of liver is the liver function as a reservoir of blood. It can store up to 0.5 to 1 liter of blood. Second function is the liver macrophage system serves as a blood cleansing function. It will purify blood, means it will filter blood. So liver macrophage system serves as a blood cleansing function. So blood flowing through the intestinal capillaries. Blood flowing from the intestinal capillaries picks up many bacteria from the intestine, and the Kupffer cell that is lined in the hepatic sinusoids, it will engulf bacteria. Or it will cleanse blood as it passes through the sinuses. Again, just repeating it. One function is the liver functions as a reservoir of blood. It can store about 0.5 to 1 liter of blood in the hepatic sinusoids and hepatic vein. Second function is the liver macrophage system. Liver macrophage system serves as a blood cleansing function, means blood filtering function, because liver sinusoids are lined by the Kupffer cell, and this Kupffer cell, it will engulf bacteria and it will ingest all the foreign matter. So it functions as blood cleansing function. Third function is the secretion of bile. One of the primary function of liver is the secretion of bile. And what is the function of bile? Main function is digestion and absorption of fat. Main function of bile is the digestion and absorption of fat through bile acid. We will study more detail about the bile and later on. So one of the primary function of liver is the secretion of bile, and bile function is it helps in the digestion. And absorption of fat. It will remove, or it will excrete excess of the bilirubin. That is produced by the destruction of RBC, and excess of the cholesterol. Excess of the cholesterol is removed through bile. Again, I am repeating it. One of the primary function of liver is the secretion of bile, and function of bile is the digestion and absorption of fat, and digestion and absorption of fat. Other is the it removes excess of the bilirubin, that is bilirubin is produced by the destruction of RBC, and excess of cholesterol is removed through bile. And fourth function is the metabolic function of liver. Metabolic function of liver. Metabolic function of liver on carbohydrate metabolism, on protein metabolism, on fat metabolism. So all these food products are metabolized by the liver. So one of the function, that is metabolic function of liver, metabolic function of liver on carbohydrate, protein, and fat. So now let us see about carbohydrate metabolism. Liver can store large amount of glycogen. Liver can store large amount of glycogen. This extra amount of glucose is converted into glycogen, and it is stored in the liver. So in carbohydrate metabolism, storage of large amount of glycogen, extra amount of glucose is converted into glycogen, and this glycogen is stored in the liver. And other function of liver is the conversion of galactose and fructose to glucose. Conversion of galactose and fructose to glucose. Glyconeogenesis. Liver function as a glyconeogenesis. Glyconeogenesis means formation of glucose. Formation of glucose from non-carbohydrate source. Gluconeogenesis means formation of glucose from non-carbohydrate source, means formation of glucose from protein and fat. Glycogenolysis is the breakdown of glycogen to glucose. Breakdown of glycogen to glucose. Now let's revise about the carbohydrate metabolism. In carbohydrate metabolism, metabolic function of liver. In carbohydrate metabolism, liver functions as a storage of glycogen. So large amount of glycogen is stored. Large amount of glycogen is stored in the liver, and liver function as a conversion of galactose and fructose to glucose. Gluconeogenesis means formation of glucose from non-carbohydrate source, that is formation of glucose from protein and fat. And glycogenolysis means breakdown of glycogen to glucose. This is the metabolic function of carbohydrate. So liver plays a major role in maintaining a normal blood glucose level, removing excess of glucose from the blood and returning it to blood as needed, so-called glucose buffering function of liver. So one of the function of liver is the glucose buffering function of liver. Glucose buffering function of liver. It means that extra amount of glucose in the blood. It is converted into glycogen and stored in the liver and muscle. Again, I'm telling, extra amount of glucose in the blood is converted into glycogen, and it is stored in the liver and muscle. And if there is low blood glucose level, if there is too low blood glucose level, then this glycogen is again converted into glucose, and it will maintain the normal blood glucose level. So liver also function as a glucose buffering function of liver. What happens if there is increased blood glucose level? One function of pancreas is the secretion of insulin. This insulin will do what insulin will do. Insulin will do, if there is increased blood glucose level, then insulin will cause the increased uptake of glucose by the cells. But liver, what will it will do that? It will convert glucose into glycogen. This liver, it will convert glucose into glycogen. Again, I am telling it. If there is increased blood glucose level, from pancreas, insulin is produced. This insulin will cause increased uptake increase uptake of glucose, increase uptake of glucose from blood into the cells, increase uptake of glucose into the cells, and liver will do that. It will convert all the glucose, it will convert extra glucose into the glycogen. So it will function as glucose buffering function of liver. So liver is the main site of alcohol metabolism, for which liver cell contain the enzyme alcohol dehydrogenase. Liver is the main site of alcohol metabolism, for which the liver cell contains enzyme alcohol dehydrogenase. In liver can metabolize alcohol. Fat metabolism. Fat metabolism. That is, liver, it will degradation of fat. Degradation of fat means emulsification of fat. Emulsification of fat is caused by the bile, and bile is produced from the liver. So in fat metabolism, there will be degradation of fat. Degradation of fat, synthesis of fat from protein, synthesis of fat from protein and carbohydrate. Synthesis of fat from proteins and carbohydrate, synthesis of large quantities of cholesterol, phospholipid, and most lipoproteins, synthesis of large quantity of cholesterol, phospholipid, and lipoprotein, beta oxidation of fatty acid, to supply energy for body function. In fat metabolism, liver functions as a degradation of fat, that is emulsification of fat by the bile, synthesis of fat from protein and carbohydrate, synthesis of large quantity of cholesterol, phospholipid, and lipoproteins, beta oxidation of fatty acid, for supply of energy. In this beta oxidation, what happens that in this beta, you have studied in the biochemistry also. In this beta oxidation, fat is converted into fatty acid and glycerol. This fatty acid this fatty acid is converted into acetyl coenzyme A, and this acetyl coenzyme A enters into the citric acid cycle and for the production of energy. So in fat metabolism, there is beta oxidation of fatty acid. In protein metabolism, deamination of amino acid. Liver is the main site of urea formation for removal of ammonia from the body fluid. Because ammonia is a toxic substance. So it must be converted into non-toxic substance, that is water soluble substance, that is urea. So liver will convert ammonia to urea, and it is removed through bile. And formation of plasma proteins. One of the function of liver is the formation of plasma protein, that is albumin. Formation of plasma proteins. So liver is the main site for formation of all non-essential amino acid by transamination of keto acids. So liver is the main site for formation of all non-essential amino acid by transamination of keto acid. Detoxification and protective function. Liver detoxify many drugs. Several of the hormones secreted by the endocrine glands are either chemically altered or excreted by the liver, including thyroxine, all steroid hormones such as estrogen, cortisol, and aldosterone. So liver damage can lead to excess accumulation of one or more of these hormones in the body fluids, and therefore cause overactivity of hormonal system. It simply means that excess of the hormones, mainly thyroxine and all steroid hormones such as estrogen, cholesterol, aldosterone, it is removed by the liver. If there is liver damage, then these hormones, then there will be excess of these hormones, and it will cause overactivity of the hormonal system. So liver also removes excess of the hormones, excess of the endocrine hormones, mainly the thyroxine and all steroid hormones. And liver liver, it also detoxify many drugs. Synthetic function. What are the synthetic function of the liver? One of the synthetic function is the liver synthesized plasma proteins. Liver synthesized plasma proteins. Main plasma proteins is the albumin. Plasma proteins are albumin, globulin, and fibrinogen. Main plasma proteins is the albumin. Albumin maintains the plasma oncotic pressure. Albumin maintains the plasma oncotic pressure, and it will synthesize some blood coagulation factors. Liver synthesize some blood coagulation factor such as fibrinogen, prothrombin, factor 7, 9, 10. That is vitamin K dependent. Vitamin K dependent clotting factors are synthesized by the liver. Vitamin K dependent clotting factors are synthesized by the liver. Most of the coagulation factor, accelerating factors, globulin, fibrinogen, and prothrombin, factor 7, 9, 10 are synthesized by the liver. Protein C, protein S, antithrombin, it is synthesized by the liver. We have already studied about protein C, protein S, antithrombin, it is synthesized by the liver. Enzymes like alkaline phosphatase, SGOT, SGPT are synthesized by the liver. Urea, liver will convert ammonia, that is toxic substance, into the water soluble non-toxic substance urea, and ultimately removed through bile. So liver will synthesize urea. Liver will synthesize cholesterol. It also synthesize cholesterol. It will synthesize insulin IGF-1. IGF-1 is also synthesized by the liver. Insulin, like growth factor 1, is synthesized by the liver, which helps in the growth of the child. Again, repeating it, synthetic function of liver. Liver will synthesize plasma proteins, that is albumin. Albumin will maintain the plasma oncotic pressure. It will synthesize most of the blood coagulation factor such as fibrinogen, globulin, accelerating factor, prothrombin, factor 7, 9, 10. That is vitamin K dependent clotting factors are synthesized by the liver. It also synthesizes protein C, protein S, and the thrombin, enzymes like alkaline phosphatase, SGOT, SGPT, and it will convert ammonia to urea. It will synthesize cholesterol. It will synthesize insulin like growth factor 1, which helps in the child growth. Storage function. Liver stores glucose. Liver store glucose in the form of glycogen. It will store vitamin B12, vitamin A, vitamin D. And liver will store iron. It stores 60% of the excess of iron, mainly in the form of ferritin. Again, liver storage function, liver stores glucose in the form of glycogen. It will store vitamin B12, vitamin A, vitamin D. It will also store iron. 60% of the excess of the iron is stored in the form of ferritin. It will store copper and vitamin K. Excretory function means these are the substance that is toxic to the body. So excretory function of liver, it will excrete dyes, certain dyes, exogenous dyes, and that is in the colored food, that we will eat in the colored food, like in the chow mein and the colored jalebi, that can by the liver. Bilirubin is the excess breakdown of RBC. This bilirubin is also toxic. It is removed by the liver. Urea is removed by the liver. Other function are the erythropoiesis. Liver is the main site of erythropoiesis in the fetal life. By secreting erythropoietin, liver also secrete erythropoietin. So erythropoiesis, liver is the main site of erythropoiesis in fetal life. Erythropoiesis is the formation of RBC. Erythropoiesis is the formation of RBC or blood cells. By secreting erythropoietin, it helps in the formation of RBC in fetal life. And liver, it is also the main site for destruction of RBC. So these are the functions of the liver. Again, we'll just revise it. Function of liver, because in your exam it is always asked about, just enumerate the, if it is of 1 mark, then there will be questions like this: enumerate the function of liver. If there is enumerate the function of liver, then you will just jot down, or you will just write down the in one line all the functions of the liver. If it is of 2 marks or 3 marks, then you will elaborate the function of liver. So let's revise the function of liver. So liver functions as a reservoir of blood, because large quantity of blood, about 0.5 liter to 1 liter of blood is stored in the hepatic sinusoids and the hepatic vein. Liver macrophage system serves as a blood cleansing function, means it will filter the blood, means all the macrophage, all the bacteria are engulfed by the macrophage, that it lines the sinusoids, and it will engulf the bacteria and foreign matter. So liver macrophage system serves as a blood cleansing function. Blood cleansing means it will filter blood. Secretion of bile. One of the primary function of liver is the secretion of bile, and function of bile is the digestion of fat and digestion and absorption of fat. We'll study in detail about the bile in later on. So one of the function of liver is the secretion of bile, and bile, it will help in the digestion and absorption of fat. Metabolic function of liver. In metabolic function of liver, it will metabolize carbohydrate, protein, and fat. In carbohydrate metabolism, it will store large amount of glycogen. It will convert galactose and fructose to glucose. Gluconeogenesis means formation of glucose from non-carbohydrate source, means formation of glucose from protein and fat. Glycogenolysis means it will break down glycogen to glucose. So liver plays a major role in maintaining a normal blood glucose level, removing excess glucose from the blood and returning it to blood as needed, so-called glucose buffering function of liver. So liver functions as a glucose buffering function. Excess glucose is converted into glycogen, and when there is a low blood glucose level, then this glycogen is again converted into glucose. So liver is the main site of alcohol metabolism, for which, because liver cells contain enzyme alcohol dehydrogenase. Fat metabolism, degradation of fat means breakdown of fat into fatty acid and glycerol by bile acid. Synthesis of fat from protein and carbohydrate. Synthesis of large quantities of cholesterol, phospholipid. In fat metabolism, liver functions as a degradation of fat by bile acid. Bile acid, it will degrade fat, means it will break down fats into fatty acid and glycerol. Synthesis of fat from protein and carbohydrate. Synthesis of large quantities of cholesterol. Cholesterol is synthesized by the liver. Phospholipids are synthesized by the liver, and lipoproteins are synthesized by the liver. Beta oxidation of fatty acid. You have studied in the biochemistry. Beta oxidation means that fat is degraded. Fat is degraded into fatty acid and glycerol. Fatty acid is converted into 2-carbon chain, that is acetyl coenzyme A. Acetyl coenzyme A enter inside the citric acid cycle for production of energy. And protein metabolism, deamination of amino acid. Deamination means removal of one amino group from amino acids. And liver is the main site of urea formation for removal of ammonia from the body, because ammonia is a toxic substance. That ammonia is converted into water-soluble substance, non-toxic substance, that is urea. And this urea is synthesized by the liver. Formation of plasma proteins, mainly the albumin. So all the plasma proteins are synthesized by the liver. Except gamma globulin. And liver is the main site for formation of all non-essential amino acid by transamination of keto acid. You'll just remember, or you just learn by heart about the function of liver, because it is asked in the exam. And this albumin, it will maintain the plasma oncotic pressure. If there is low plasma proteins, then there will be swelling in the periphery. Detoxification and protective function. So liver detoxify many drugs. So several of the hormones, mostly the endocrine hormones, mainly the thyroxine and all steroid hormones such as estrogen, cortisol, and aldosterone. If these hormones are in excess, it is removed by the liver. If there is liver damage, then all these hormones will be in excess, and it will cause overactivity of hormonal system. Synthetic function, it will synthesize plasma proteins, that is albumin, some blood coagulation factors like fibrinogen, prothrombin, globulin, and acylating factors, factor 2, 7, 9, 10, that is vitamin K dependent clotting factor, protein C, protein S, antithrombin, enzymes like alkaline phosphatase, SGOT, SGPT, and it will convert ammonia to urea, liver will synthesize cholesterol, it will synthesize insulin like growth factor 1, that is important in child growth. Storage function, liver store glucose in the form of glycogen. So liver can store glycogen. It will store vitamin B12, vitamin A, vitamin D. Iron is stored. Iron is stored mainly in the form of ferritin. It will store copper, vitamin K. Excretory function. Excretory function is the toxic substance that is produced in the body. Toxic substance that is produced in the body are excreted through liver. Other excretory waste products are also removed from the kidney through urine. So certain exogenous dyes uh, such as bromosulfatylene and rose bengal dye means all the colored substance that we consume is excreted by the liver. Bilirubin that is produced from the RBC breakdown is removed by the liver. Urea, that is ammonia, is converted into urea, and urea it will secreted by the liver and it entered into the bile canal fluid. So bile canal fluid, urea is either excreted in the urine or in the feces. Other functions are the erythropoiesis. So liver is the main site of erythropoiesis in fetal life and destruction of RBC. So these are the main functions of liver. If it is of 1 mark, then you will just write down in 1 line about the function of liver. If it is of 2 marks or 3 marks, generally it is asked in the SAQ and the PBQ. Then you will enumerate all the function of liver. So in the next class, we'll study about we'll study detail about the bile and how the bile is formed. So one of the main function of liver is the secretion of bile. And this bile, it helps in the digestion and absorption of fat. So how bile will help in the digestion and absorption of fat, we'll study in the next class. Just summarizing it about the function of bile. So bile serves 2 important functions. Bile plays an important role in fat digestion and absorption. Bile serves as means for excretion of waste products from the blood, bilirubin. That bilirubin and excess of cholesterol is removed through bile. If you have, okay, we have finished the function of liver, and next class we'll study about the synthesis of bile and how bile is synthesized. If you have any question, then you can free to ask.